everyone. My name is Samantha Maragh. I lead the genome editing program at the U.S. National Institute of Standards, and I'm going to tell you a bit about this consortium that I run and this Interlab study that we have done in the area of technologies that are of use and interesting for genome editing variant detection. So just one disclaimer statement. I'm going to tell you about some instrumentation technologies in this study. Those are not to be interpreted as endorsements by NIST of any particular technology. So I just told you that I'm from NIST, the U.S. National Institute of Standards and Technology. You may have heard of NIST. It's more likely maybe you haven't heard of NIST. So let me tell you who we are. So NIST is a part of the U.S. federal government. We're part of the Department of Commerce. We're non-regulatory. We are non-funding. Our primary purpose isn't to fund. Our purpose is to help you know whether the measurements, the standards that are needed to know whether technologies that you're using are giving you the right answer. So to enable you with the power of knowledge so that you can know whether you're getting good data and you can make important decisions based on that data. NIST's scope is any and everything that can and should be measured. And we are your metrology institute. We work with other federal government agencies, including regulators like the FDA, to help them know whether the data and the products that they're regulating, that they can trust that information. So for today's topic, I'm going to tell you a bit about the work that we're doing at NIST in standards for genome editing. And so just to level set the audience, let's take a brief overview of when I talk about genome editing, this is really what I'm talking about. You have some initial reagents that are going into this process. So genome editing molecules, there are a variety of them. Many of you may have heard of CRISPR or CRISPR-Cas systems. That's a very popular system. There are other systems as well. And in general, these systems do something to damage or manipulate DNA. You've got to be able to get those systems, deliver them into your cells of interest. One of the key things about genome editing technologies is that you can make DNA sequence changes in live cells, and those cells are still functional and able to do a purpose after that editing. So you're getting them into your cells. Hopefully you get a DNA sequence change. Hopefully you get a DNA sequence change that is meaningful for you. And that those cells would then be engineered. In this specific case, if you're using editing tools, they'd be edited. And then you've got to understand if they're fit for your purpose. And the purposes of genome editing can be multifold. There's a lot of emphasis just right now on the first gene therapy using CRISPR as the modality for sickle cell disease. But there are a variety of applications for genome editing where you might want to be able to intentionally and specifically manipulate the genetic sequence of a cell so that it does something that's meaningful for you. So I will tell you a bit about my program and sort of where this all fits into the things that you all might be interested in or want to know about. So I lead a program at NIST, a genome editing program, and that vision is really just to enable all of those products that might use genome editing as their underlying modality to be able to accelerate their process to market. And the way that you would do that is increasing the confidence of knowing whether you understand whether the genome editing tool has acted in a way that you need it to. And so some of the ways that we think about this is that you might need physical samples as you're doing a genome editing experiment or evaluating the technologies that you use to understand your samples. You might need physical kinds of controls. You may need data sets, that sort of thing. Data and metadata, you might need to understand the data sets, have the benchmark data sets, have ways of describing the initial study in terms of metadata, so that goes along with that data set to make sure that it's usable. And documentary, what are recommended practices? Are there standard definitions for things? What can be used as documentary norms that can help the community do things in a way that's reliable and standardized? So the primary use case in the work that I do at NIST right now is being driven by this gene therapy use case. And it's an extremely exciting time in the genome editing field, as we've just had the first human therapeutic approved by FDA based on using a genome editing technology, specifically CRISPR. So there are different ways of doing this. As you look at this, you can take cells from a patient, you can engineer their own cells and put them back into them. That's the process of this new Cascavy drug. Or you can have allogenetic, where you take something from a healthy donor, you engineer those cells, and those cells are now able to be medicines for patients that have some sort of illness. Or you can think about in vivo delivery, where the tissue that you might need to have a therapeutic impact on may not be something you can take out. Maybe it's brain or neural tissue, maybe it's heart tissue. And so you really need to be able to get those biomolecules into that person, into those cells, and hopefully they do what they're supposed to do. And so that might be the modality. So all of these things are areas that people are looking to know, how do we know that the measurements that we're using to understand our genome editing are giving us good information? And in context here, FDA requires reporting of what's happening here. So on target, you intend to change a specific location because it's meaningful for you in a certain way. What have you done at that location? Off target, maybe this process, I'll just tell you right now, it's not perfect. It makes mistakes. It makes changes at places in the genome that you didn't intend. That's called off target. So where in the genome did those changes happen, and what happened at those genomic positions? And the relative frequency of what's happening in these pools of cells. And I'll show you a little bit more about that in a slide or two. So we have to define the problem. And I want you to think about this in general, not just if you're talking about genome editing, but in general, as you do your science, I really want you to think about these questions. Where in your process is it very important to know if you have the right answer, your data is reliable? Where of these processes is it currently difficult to understand if the answer is reliable, and what can be done to give more confidence? So I want you to be thinking about that, because I want you to have confidence in every aspect of the science and the work that you do, and really asking yourselves these questions can help give you that measurement science mentality as you go through all of the things that you're doing. So where do we start? What are the needs for the genome editing field? So at NIST, we had several engagements several years ago with the genome editing community to say, asking those questions on the previous slide, where do you have needs? What's challenging? And here's some of the key takeaways that we got from those conversations. You're starting with genome editing molecules, and the question is, how do we understand whether the quality of these genome editing molecules, are there norms there? So that was an important thing that came out. We need to deliver things into cells. Are the delivery systems working? Are they robust? How do we characterize them? That came out of this. One of the things that we don't talk about a lot in a broader sense in the community, something that we talk about a lot in specifically the genome editing, people that are doing the editing, is that these editing molecules, they damage DNA, they make breaks, they make nicks. So where is this happening? What off-target locations might you have this activity at? And ultimately, you're making DNA sequence changes. And as I'll show in a follow-up slide, these may be pools of cells. How do you know whether you're actually detecting correctly and reporting correctly the variety of edits you might have on and off target? And so that's actually will be the focus of what I'll tell you a little bit more about some of the work that we've done starting at that particular problem, because that's usually what you're using to interpret a lot of what's happening upstream. And if you don't know if you're getting the right answer there, it's very hard to understand whether your delivery system or your reagent had some sort of impact. So as we look at what's going on at NIST and the editing process I showed you about before, there's tons of innovation happening at the top. At NIST, we think about the measurements and the control samples. That's something that we can provide. We can support with data and control data sets, and supporting with metadata norms that people can use to capture the information about their studies. One of the ways that we're doing this is that I lead a genome editing consortium. And that consortium has now almost 50 organizations that are members. The mission of that consortium is to really be able to have a mechanism to gather all of these different kinds of organizations together under one umbrella that allows them to work together, as well as working with NIST, to try to develop some of these control strategies, some of the tools that would be helpful across the community. And the way we do that is that we have a working group that's really focused on the physical measurements and assays, a working group focused on data and metadata, and a working group focused on lexicon or technology. That'll look very similar to the way that I've designed the program in general. And so over here is the list of members of the consortium to date. So one of the things that I've mentioned in previous slides is this idea of sort of walking you through the complexity of genome editing. And these outcomes can be very complex because while these approaches are targeted, we can't control at this current state of the technology's development, we can't control the outcomes of what edits get generated. We can try to influence that by providing donors, but we can't really control the whole process. So just as a model here, you might have a set of cells that's unedited. You're going to design editing molecules, deliver in, and you might have unedited cells. You might have cells that have the edits you want. You might have cells that have edits you don't want, and cells that have both of those things, edits you want and don't want. Probably going to take subsample some of that, and you're going to do some sort of analysis. You're going to do some DNA analysis to try to understand what variants you made. And over here with the sequencing, this is actually a set of real data from an experiment from the consortium, where this is at the on-target site, and you have this array of different alleles that are generated. And one of the questions is this correct, right? I didn't have a control. I don't know what I should expect. Is this correct? And what's not shown here is the possibility of larger variants. And so there's the schematic here shows that you can have larger rearrangements. That is a possibility. And so you need to know if those larger variants did happen, do you have the ability to detect them if they were present? And there are a lot of challenges with this. And so one of the things that came out of this is that it'd be really great if we had some initial control samples that allowed us to understand whether the approaches of DNA detection that people are using are giving the right answer. And so we started into an initial project, a mixture study, to begin our first step at trying to get some samples that have key characteristics that could be used across different DNA detection assays. And so these are controls with relevant characteristics. Where do we go for these controls? Ultimately, what we're working on, and I won't really tell you about that today, but we're working on actually engineering a bunch of variants at the same genomic locus so you could have that allele pileup like you saw on the previous slide. But in the absence of having that immediately accessible, what we could get our hands on are these genome in a bottle samples that are very highly characterized, that we knew the entire genomic sequence with a lot of confidence, and that we could use to make blends of different people's genomes and get at some of these questions. And so if you're not familiar with the Genome in a Bottle program and you do any sort of DNA detection, I highly encourage you to look up this program. This is work done by NIST, led by Justin Zook, that has really revolutionized DNA detection. And I know some of you are here to hear more about, Bionano's technology. Bionano was a key part of this analysis as well with this Genome in a Bottle program. And so we have these samples that are very highly characterized. The next thing is that what did they need to be able to do? So we have this consortium, and the consortium members are there as the experts in doing these sorts of technologies and trying to make product. And so we defined together some key benchmarks that the samples needed to have. So we said, well, there was what I would call identity, both length and sequence, benchmark saying that, yes, these samples have these lists of the variants that people wanted as benchmark sizes, and that we know what the sequence is base for base of those, and that we were providing them at a few different frequencies. So there were 5 set of samples, and the samples had DNA. We made them as DNA mixtures because some technologies needed DNA. Other technologies, Bionano was a participant in this work, and their technology as well, as well as other technologies needed cells as the starting material. And so we actually had to take a step back and really think about how are we going to mix cells such that we felt confident that we were getting these allele frequencies and fractions. And that's something that we developed a process for at NIST. I don't really have the time to tell you about that today, how we did that, but it is a process that we now have that we feel comfortable that we were able to make those sorts of mixtures. And we made those samples as cell-based mixtures, DNA-based mixtures, and gave them to Interlab participants. And so the Interlab participants, here's a high-level overview of the data that we have in to date, the kinds of technologies that people are using. It's very difficult to see here, but here's Bionano's optical genome mapping technology. And for the data that we have in currently, they are the only technology that it's a genome-wide technology. So that was a very valuable data set to understand what's possible without having to target. So what I will show you here is that here are participants in this Interlab study. They're technology makers. These are organizations that make a technology. They will be identified with their data sets. And technology users, these are organizations that, I would call NIST a user. We're just trying to onboard somebody else's technology and use it to the best of our ability. They are not going to be identified with their data sets. They're going to have a description of what their workflow was. Metadata and data return. So at the beginning of this study, we identified a set of metadata that everybody would need to return. And then we said everybody's going to give this information back, these data sets back, raw and processed, so that this could all be used by others in the Interlab study and then made publicly available as we will publish our results and all of the information related to the samples. This is just a brief, just to give you a sense of where some of the technologies that were part of the study sit, whether they are using NGS sequencing or not, kinds of approaches. Okay, so we are at the point of this Interlab study that we have data. And so I'm going to give you a little bit about some of that data now. We had some key questions that we wanted to discuss in the Interlab study. Were the mixtures what we expected them to be? Workflows, additional variants. And so I'll give you just a little bit of what's going on there. And we'll be primed to put together a publication right now that we've started to work towards. First thing I want to let you know is that, yes, the variants that we attempted to have present were both present, as well as present at the mixture fraction bins that we verified that we would have for the Interlab samples. And so Illumina got DNA-based mixtures as well as the DNA extracted from our cell-based mixtures. They designed a custom panel along with Mission Bio that does single-cell sequencing. They designed a custom panel for the cell-based mixtures, NIST did digital PCR, and we verified the mixture frequencies and compared that to the sequence-based detection to verify all the variants before the consortium members got them. So one of the things I want to point out here, one of our big takeaways is the bioinformatic challenge and burden when you're doing NGS. And so what you'll see here is in the shaded bars, you see what the Illumina Dragon pipeline reported in terms of the percent of the variants that they were supposed to be able to call for each of the 5 frequency bins. The 0 bin, they correctly did not identify any variants. There were none present. But the dotted lines are showing you that if you go in and you manually inspect, that 100% of the variants are detected. And so this gap between shaded and dotted line is the gap between what was present in the dotted line that's confirmed visually manually versus what the pipeline in the iteration that it was at was able to report, which was not benchmarked for large variant detection. And so that's just something to just keep in mind, that your pipeline, your workflow, it will tell you about what it's capable of telling you about, and there may be some challenges there. And this was a general theme that we found, that there are bioinformatics gaps and differences between what can be called versus what you could visually confirm in manual inspection. So this is just to show you a bit about what the technologies said they could report in terms of their sizing. For Illumina and Mission Bio, they were unblinded. So they knew what they were supposed to find. They were control with NIST. So we sort of shaded that, yes, normally they're more, in this a few 100-ish, 100, 150 maybe, but they got additional capacity. These other technologies that are used, these were based on the organization, their documentation, the reporting of what they might be able to detect in terms of sizing. I'll point out here that we have 3 technologies that were large variant technologies, and I'll focus a little bit more on those so you can clearly see the data from those, because that's an area that we really want to know what the capability is in this community. And so optical genome mapping was the only genome-wide technology in these large variants, and the other 2 were targeted. One was an NGS-based, and the other was imaging. Just to give you a sense of this, you'll be able to go back and take a look at this in more detail than I'm putting the slides up for. But one of our takeaways is that no single blinded workflow was able to detect all of the variants. And so when I say blinded workflow, all of the participants and all of the technologies below this dotted line, they were given a list of genomic coordinates. They did not know what variant or what frequency they should find at that location. And that was part of this analysis. And that's very similar to what you'd have in a real genome editing study. You've got a location that you care about, but you don't know what you should find there. And so not all, if you look across, not all of these technologies found the same found all of the variants. However, we're able to find all of the variant bins, all of the variants in at least one technology. So this reinforced the idea that complementary combinatorial approaches to analysis are really powerful and maybe the way that people should think about things. And so down here at the bottom are our larger variant technologies, and you can see that they are pushing towards the larger variants, where most of the other short read Illumina NGS-based technologies are not able to see those sizes. Just pulling out the large variants, you can see that that as the sample frequency decreases, that you do lose the ability to detect. That's very much what we expect. It's the same trend with NGS-based technologies, but just pulling out that the large variants are detectable, even very well detectable down to 7.5%. We don't, the next jump down is 1.25%. Still some things detectable and just well below what we thought anybody could detect. We still had at least something detected at 0.125%, which wasn't expected at all. So just for some of the variant allele frequencies, just to give you a sense of this, that there was a lot of spread, even with technologies that had short read capability. As you got into even as small as like less than 15, you got a big spread in terms of the variant allele frequencies that you were seeing. One of the things just to note here is that while there was spread, the variants that were detected by large variant technologies, now again, not all of them reported, but those that did report, they were generally close to the benchmark values, which was again very impressive in terms of the ability to detect larger things and also quantify. But it was very unexpected that even as you got to even one base pair, just the spread you started getting in the frequencies being detected being outside of what was expected, or a broader and broader spread, what was well within the NGS detection limit. So I'll just take 1 minute and show you a few other key takeaways. Some variants were repeatedly overcalled by multiple pipelines. And so we're looking into this. Why might that be? Is there something that we can learn about the characteristics of these variants? Some variants are repeatedly undercalled by multiple pipelines and workflows. And again, similarly, we're trying to understand if there's something we can learn, some trend, something to come out of what we can learn about this. We found that we did something that was a lot of work, but we thought would be of good value to the community, is that NIST went and manually inspected everything that was reported. And we found that for NGS-based reporting, that about a third of variants that were not called, NIST provided the variant and the verification, but we didn't get reporting on these from the participant, that when NIST went back and manually inspected the raw data, that around a third of them were present visually, but weren't called by the bioinformatics. And so this is, again, one of those things to consider about how do we improve, how do we think about some of these false negatives. So one other thing is that some variants were visible, as I was just showing you, but posed repeatedly for more than one workflow bioinformatic challenge of resolving it. You could see it and it called something and it just wasn't the correct thing. And we found that that there were some things where there was intermolecular similarity or very close similarity right upstream or downstream that might be confounding some of the alignment algorithms. So again, for the kind of analysis that needs alignment, these are some of the considerations and things that we identified in this study. So just in conclusion, some takeaways here that you can take a look at. I won't read through all of them as I just went through them. But I want to tell you about some of the next steps now that I've just given you a brief overview of what's going on. So analysis of additional variants. We're looking at that sort of positives that were reported that may or may not be false positives. What are those? What are their characteristics? We have an additional technology that wasn't shown, a BioScribe, that they were blinded for single cell whole genome sequencing, seeing how that data set merges into what we have already. We're working on a manuscript where all of the information, the data, the metadata, the data sets will become publicly available. And working on a phase 2 set of clonal engineered samples that would actually have all those kinds of variants and alleles at the same genomic coordinate. It's a true more similar to your real life situation control for the genome editing DNA detection challenge. So I want to say thank you to all the Genome Editing Consortium members and other collaborators and my NIST colleagues that were part of this work. If you're interested in hearing more about this or potentially joining the consortium, please feel free to reach out. Thank you.